So let's continue with the final part of this block of this segment of our agenda. So we have Addison Carroll next from the University of Nebraska Lincoln. Allison is going to speak to us about her research on the use of different byproducts with regards to dairy cattle. Go ahead, Addison, the floor is yours.
Everyone. My name is Addison Carroll, and I'm a PhD student at the University of Nebraska-Lincoln working with Dr. Paul Kononov. My area of focus is lactating dairy cattle nutrition as well as energy metabolism. I am so excited to get to the opportunity to talk with you all today, so let's get into it. Let me share my screen real quick. All right. So today I am coming to talk to you all about from feed preference to energy partitioning, feeding co-products to lactating dairy cattle. So kind of to begin, I wanted to give you all an idea of the dairy industry today. In 2020, fluid milk provided enough energy, protein, and calcium to meet the needs of 71, 16, and 245 million people. Now, the dairy industry is kind of tough financially. And in 2021, the USDA estimated that production costs would outweigh milk profit from farms that span from under 50 cows all the way over to 2,000 head. And on the right is actually a farm that I worked on myself that had about 100 head. So this hits home for me a lot. And so the question is, how do we deal with these tight margins, right? What can we do? What strategies can we utilize? And what different types of products can we utilize in our rations to make these girls produce better and help our farmers create some profit? So the beautiful thing about ruminants are they are able to convert fibrous human inedible inputs and create these human edible outputs. So the question is today, where do co-products fit in this whole situation from animal preference all the way through energy partitioning? So I love to talk about this figure here, feeding co-products contributing to a better planet. So really, when we look at co-products, they just span the whole gambit, right? We feed them to the animals, and then they're able to produce food and fiber and go back into the people and industry. Maybe they produce fertilizer and all of these things, and they come together to make this really holistic product. So I do have a question though. So in models and in nutritional studies, we generally see increasing intake with increasing distillers grain inclusion. Now, just kind of take a second and think about why that may be now. So in models, they would say, well, the DDGs improve digestibility. Maybe they reduce the NDF to ADF ratio. Maybe they provide sufficient protein to maximize that MCP or microbial crude protein production. However, I would like to ask a question. What about palatability? What about preference? Like for me, I love ice cream so much, and I dislike tomatoes very, very much. I'm sorry. But we think about as humans our preference and what we enjoy all the time, but in cattle, we don't. So cattle have approximately 20,000 taste buds on their tongues, and they're highly sensitive to flavor distinctions, but there's been very little exploration into what they may like. So in order to kind of test what these animals like, we actually do a test preference experiment at the University of Nebraska-Lincoln. Today I'll be talking about 2 experiments. And so, as you can see here, we have our first experiment, we fed 1.13 kilograms. In our second experiment, we fed 0.5 kilograms. And what we do is we lay out these feeds or these pellets in the second experiment in front of the animal, and we let them make a choice. So we offer them 4 feeds for 4 days, and then we tally up and we see which one do they really like the most. And then we rank that as number 1 and we take it and pull it away. So then we offer them the next 3 feeds for 3 days, and then we pull the most preferred. And then the last are 2 feeds offered for 2 days. And that's how we're able to really make a sequential ranking of these different feeds. Now, I wanted to show a quick video here of what we see in an animal. This was actually from a study, the second experiment that I'll be discussing today. So as you can see, we place these bins in front of this animal. She's able to kind of munch and taste and test. And you may ask, well, does where the bins are placed influence what she prefers? And I would actually say no, because I've seen animals reach all the way to the left or all the way to the right just to get something that they enjoy eating. So I get that question a lot and just wanted to go ahead and discuss it while we were here. So in our first experiment, this is a feed preference test results between, we're looking at corn, dried distiller's grains, an animal protein, and an equal blend of all three. Now, over here is our cow ID, and then our different treatments across the top. And we have these numbers as their rankings, right? So if it's a 1, they liked it the most, 2, 3, 4, and so on. And as we can see from this experiment, those cows really enjoyed consuming the distiller's grain treatment, right? There was only one animal who did not or chose it as third. So that's really promising. And so with that, we were able to analyze the data and say, you know, the probability this animal would choose those distiller's grains over other feeds was about 90.6% of the time, which is really interesting. So then we're thinking, okay, co-products, you know, where are these things fitting in? Where can we use this? And we actually did another experiment not too long ago this past year where we looked at pellets, right? Because in dairy cattle nutrition, we're seeing more and more use of automated milking systems. And so to get an animal to go to the milking system, we have pellets that we offer kind of to incentivize her to go. So we saw, okay, you know, what can we put in these pellets to see what's going on? So we have all of our animals here. And then our 4 treatments were a corn gluten feed pellet, which was super easy for us to obtain in the Midwest, relatively cheap. The next one was called Flavor. And so that had an oregano leaf, it had a distiller's grains component, a corn component. These are all things that we've observed to be highly palatable to the animal. So we kind of wanted to give something that packed a punch in terms of taste. The next one was ENG, which had a high percentage of ground corn. And the last one was C mix, which was like a general concentrate mix, which included both the vitamins and the minerals. So what we observed, right, was that animals really enjoyed the corn gluten feed. They really liked it. It was really palatable to them. Once again, we only see that 2 animals chose it second. And then our next was this flavor. So, you know, we started thinking about what could be going on with these animals, what could be driving these animals towards co-products, right? So I think in my mind, I have 3 things that I've pinpointed that might be what's pushing those animals towards those products. And that for me, the first thing I think about is the Maillard products, right? During heating process, these create Maillard products, which are kind of those caramelized flavors and scents you get from like when you cook a steak or something like that. Also, there's a yeast component. And so like nutritional yeast has this kind of cheesy flavor. And then the last one is glutamate, right? MSG. And we've actually seen in a study that animals like MSG. And so I think our goal at UNL in the future here is to kind of try and divide out, you know, the parts of distiller's grains that, you know, we think might be driving this preference and see, you know, is there a specific thing, or does distiller's grains just kind of bring it all together in a perfect combination? So now I'm going to move into kind of our larger study, a little bit more in depth, a little bit more kind of rigorous experiment. And so the title is Energy Utilization of Lactating Dairy Cattle Fed Increasing Inclusion of a New High Protein Co-Product. I conducted this during my master's here at UNL, and I'm discussing it with you all today. So when we think about new feeds that come into the market, now I started my master's in 2020, and at this point in time, there was not much or at all of an exploration into these new high protein co-products. And so on the screen, I have a ration formulation software, and I just started typing in, you know, high pro, corn distillers, all of these things, and I could not find a high protein, right? But as of yesterday, I actually found a high pro distillers grains, but there's only one. And these products are very diverse and very different. And so as a result of this, we have this high protein co-product. You know, the problem is data is limited on the chemical composition and how animals utilize it. And then we have no data in the feed library, unknown animal performance, and then thus limited ration formulation strategies and optimal ingredient inclusion. Now I say optimal loosely because optimal can mean different things in different production situations. So I recognize that. And so the first objective I will not be covering today, but the second objective I will be. So the objective of the second experiment was to examine the effects of increasing inclusion of the new product on whole animal energy utilization. So just a little bit of background into energy utilization. The energetic efficiency of milk production is between about 22 to 34% of the total energy an animal consumes, which ultimately means that we are losing a lot of the energy that animal eats to the environment as heat, as fecal waste, as urinary waste, all of these things. So for greater utilization of this product in the field, we need controlled feeding experiments with differing feeding strategies to determine kind of the inclusion, the optimal inclusion of these new products. So therefore, our objectives were to examine the effect of replacing non-enzymatically browned soybean meal with a high protein co-product on dry matter intake, energy and nitrogen utilization, and production of lactating Jersey cattle. We hypothesized that the new high protein co-product would perform similarly in an isonitrogenous ration compared to the non-enzymatically browned soybean meal. So a little bit about how we do this here at UNL. This figure is kind of going to walk through our procedures. So what we had was 12 multiparous lactating Jersey cows. What we did is we brought them in for a period of time. We let them milk, let them get acclimated to our system, and we blocked them by milk yield and randomly assigned them to one of 4 treatments. Now, the 4 TMRs containing the increasing inclusion of the high protein co-product were a control, which was 0% high protein co-product, but 8% non-enzymatically browned soybean meal. Then we had a 2.6 low, which was 2.6% high protein co-product, and 5.4% non-enzymatically browned soybean meal. A 5.4 medium, which was 5.4% of the high protein co-product and 2.6% of the non-enzymatically browned soybean meal, and 8.0 high, which was 8% high protein co-product and 0% non-enzymatically browned soybean meal. For our energy balance, what we did is we took total fecal and urine collection. We also looked at their refusals, and we took 2-day milk samples. Now, this is kind of a top-down view of our animals with our cows and their fecal tubs and urinary tubs, kind of our collection week setup, as you will. Now, during a 23-hour period, we looked at continuous CO2 consumption, CO2 and methane production, as well as a composite, which was in this bag, O2 consumption, CO2, and methane production. And with that, via the Brouwer equation, we were actually able to estimate the heat production of that animal. Now, once we had all of this stuff collected, we analyzed those samples for those feces, feed ingredients, and refusals. We did urinary nitrogen analysis, and then we did FOM calorimetry on the feed ingredients and refusals to essentially get the initial gross energy value. Now, statistically, our data was analyzed with the Glimmick's procedure of SAS, fixed effects of high protein co-product inclusion, random effects of period square and count nested in square, and we tested the linear, quadratic, and cubic levels of high protein co-product inclusion. Now, so I first wanted to orient you to this diet because I think that's an integral place to start when we're looking at these different dairy experiments. Now, so I want to orient you to the diet. So we have the diet ingredients as well as their composition. And then we have the control, which was 0% high protein co-product, spanning all the way to the 8% high protein co-product inclusion, as you can see here and here. Now, I would like to note on this diet, we did have increasing total fatty acids with the increasing inclusion of the high protein co-product, just because naturally what that brings to the table versus a non-enzymatically browned soybean meal. Also, in particular, we saw an increase in the C18 fatty acids because of that corn oil. Also, to note, our crude protein content was 52.4% for the new high protein co-product. So once we know the diets, we can move into some sort of measure of digestibility. And this is a great side because there was no difference in our digestibility measures between all of the diets. So digestibility was similar from the control all the way through the 8.0 high. So that makes my job pretty simple on this slide. Next, we're going to move into the energy metabolism focus. And I thought I'd take a minute here to kind of break down energy metabolism, give you a little refresher if you need it. So ultimately, we have energy coming from the diet as gross energy, right? And that's all of those macronutrients times their enthalpy. But that's not ultimately what the animal gets, because we can lose energy in the feces through limited digestibility. We have something called digestible energy, right? So that energy has been digested, after which we can lose energy to gas or the urine as well through different processes of rumen metabolism and just animal metabolism in general. Then we have metabolizable energy, right? That's been metabolized. And that metabolizable energy then can be used for these different functions, right? So we first have to go towards maintenance energy, right? Because we have to maintain the body. But then we can look at pregnancy, tissue energy, and most importantly, milk energy. And what's produced between these two is heat, because these metabolic processes produce heat as a byproduct. So today I'm going to discuss kind of our linear effects between the control and the 8.0 high. So wherever you see a yellow box, that will be your control, which was a 0% high protein co-product. And your 8.0 high is the 8% high protein co-product. So first, we did see a tendency for an increase in gross energy content, likely due to the contribution of the fatty acids within the co-product itself. We saw a slight uptick in terms of the fecal energy output of the animals that produce or consumed the 8.0 high. But then ultimately we saw no difference in the digestible energy, just because we started off a little bit higher in terms of gross energy. So then we saw no difference in our methane energy presented here, but we did see a linear tendency for increasing urinary energy output with increasing inclusion of the high protein co-product. So for metabolizable energy, there's not a whole lot of difference here. So our metabolizable energy was actually very similar between the two diets. Also, our heat production was quite similar among our treatments. Now, moving into net energy, we can see that pregnancy energy was not different among our treatments, and neither was energy allotted to tissue. But we saw something interesting occur in milk energy, with a linear increase in milk energy from 23.7 megcals to 26.1 megcals per day for our 8.0 high. Now, when we start thinking about why this occurs, we need to think about, efficiency and why is energy being selected to be placed within the milk itself. So whenever we start looking at these efficiencies themselves, we can see that from the change from digestible to metabolizable energy was not different within our animals. That's really, I think, kind of what I would expect based on the gas and urinary energy. However, we did see an increase in terms of the efficiency at which ME was turned into net energy. Now, in my mind, that's kind of interesting because we're becoming more efficient. So we need to start thinking about what's causing that efficiency and what's driving that to be more efficient. So that's kind of where it's going right there. So we did see, as we would expect, since milk energy increased, we saw that energy corrected milk increased linear with increasing inclusion. We did see a tendency for increased protein. And then we saw this really strong increase in terms of fat percent and fat yield. Now, when we start thinking and putting these pieces together, we kind of discussed in our lab, what could be driving these things and pushing these things forward. And so at first, we look at this protein response and we think, well, maybe it has to do with the amino acids. But we looked at the NASEM kind of software and played around with amino acids, and we thought, maybe that's not the actual driver there. And we actually noted that, the driver is likely increased energy. And that's because in the new NASEM 21, we see that DE is also implemented into the protein equation as well. Now, for the fat, that's another story. So this corn oil, right, so the C18 fatty acids, whenever they're balanced correctly, they can be really efficiently placed within the milk fat, right? We can automatically utilize these long chain fatty acids within our milk fats versus having to produce it via de novo synthesis. So I think that could also be a driver with this fat response. But also, I think that there could be maybe a shifting occurring of the ratio of milk protein to milk fat. And so protein itself is very energetically taxing to produce. And so we think maybe that shift in that ratio could have also helped us produce more. So ultimately, when we look at these different things, digestibility of the nutrients was unaffected by increasing inclusion of the high protein co-product. Gross energy and megcals per day and megcals per kilogram increased with increasing inclusion, but did not affect DE or ME in terms of the megcals per day or megcals per kilogram, which I did not present today. With increasing inclusion of the high protein co-product, we saw increased energy corrected milk and milk fat production and tendency for increased protein production, likely as a function of that increasing efficiency or the NEL to ME ratio. So our overall conclusions is that animals will preferentially consume these fermented kind of co-products over other feeds, and that the new high protein co-product can effectively be utilized in ration formulation, similar to other high protein products. With increasing inclusion of the high protein co-product, we saw no difference in digestibility, increased gross energy content and intake, as well as increasing utilization of energy for NEL with subsequent increases in milk fat production. With that, I would love to thank the people that made this opportunity possible. In terms of funding, it's Flint Hills Resources, now owned by Poet, Fluid Quip Technologies, my advisor, Dr. Paul Konanoff, our barn staff, Aaron Rhodes and Darren Stridzik, our graduate students and undergraduate students who helped me with this process, as well as the U.S. Grains Council for allowing me to come talk to you all today. And with that, I'll take any questions you may have. Thanks.
Questions for Addison?
Vi que hubo un efecto.
I saw that there was an effect on the corrected milk in favor of the LDG, but I don't remember the level of fiber of neutral detergent, acid detergent, that has an impact in the gluconogenic relationship in the fatty acids, which is going to affect the corrected milk. That's the question.
I believe in terms of our chemical composition of our diets, the acid detergent fiber remained very similar.
Addison, Pedro Urriola, the chair from the session.
Whether during the preference test, whether you measure actual feed intake or only the first bucket where the cow was consuming in, and if you measure feed intake, did you compare nutrient intake from the different rations that the cows were picking in? Otherwise, are cows picking up nutrients instead of the ration?
Yeah, that's an amazing question.
Es una excelente pregunta.
Actually measure the intake response in terms of the choices. So we cannot currently attribute any type of preference to nutrients. Now, that's a great question and something hopefully we'll be able to answer in the future, because sometimes it does seem like animals choose things that have maybe more energy or more protein to meet their needs.
Y algunas veces,
And
¿no?
sometimes they trace minerals.
Mm-hmm.
Bueno, más preguntas para Addison.
Any more questions for Addison? Yes, we have another question here. Addison, in the use of DDGS that we were working with, among 24 and 28% of protein, one of the attributes is a soluble fiber. So one of the features is soluble fiber. In this case, the DDGS, high in fiber, high in protein. What can you say about the nutritional value of that fiber?
Yeah, so in terms of fiber for this high protein co-product, we're still trying to understand it actually, because NDF is a proximate assay for us. And so what we see in this high protein co-product is about 30% NDF, which may or may not actually represent fiber. It actually may be representing maybe that yeast fraction, because in other areas, such as like fecal NDFs, which have high microbial load, sometimes we'll see increased NDF values. So for the high protein co-product, they're supposed to remove some of that fiber. So the main kind of area for that product would be as a protein concentrate. Now, there are, as a part of that production process, there are other DDGs which have a greater portion of that soluble fiber. And so as a result, we're kind of pulling apart those two pieces, and then you have to decide, is the protein content more important to you, or is that soluble fiber punch more important to you in your formulation?
Bien, creo que
Yes, we have another question regarding the protein. Degrading ability of the protein will not have an effect on the corrected milk production due to the protein that's less degradable to more degradable. That's the question.
Yeah, so with this high protein co-product right now, we're also still trying to understand the degradability of the protein. So in the Ross assay, which is an in vitro assay, it shows about 80% RUP, or rumen undegradable protein. Whereas with a mobile bag assay, which I conducted in my master's, I saw about 50%. So if I were going to hedge my bets, I would say that it's maybe a little, probably in between, somewhere in between that 80% and that 60% or 50%, pardon me. And so it could influence the protein, essentially, response that we saw. However, we need to be cognizant that maybe that non-enzymatically brown soybean meal has more RUP relative to this high protein co-product.
Bueno, muchas gracias.
Thank you very much, Addison, for this very important presentation. Thank you.